Welcome to Unit 6, Lecture 8. So this lecture is going to focus on Step 2 of protein synthesis. So Lecture 7 was talking about RNA, basically, and Step 1, which is transcription. This is going to talk about Step 2, which is translation. I apologize. This is a little bit longer of a presentation, so just bear with me. Learning objectives. Explain how the genetic code works. Describe the role of ribosomes in assembling proteins. Understand how molecular biology relates to genetics. So remember, there are the three main forms of RNA. And in the second step of protein synthesis, all three of them come together to do work. So we've got the messenger RNA, we've got the transfer RNA, and we got the ribosomal RNA. So ribosomal RNA is the thing that's making up the ribosome, which is where step 2 happens. Okay, so if you can see here, this is step 2 in one little picture. Okay, there's a lot happening in this little picture, but this is all step 2, and you can see we've got the mRNA, we've got the tRNA here. And then this whole ribosomal thing is part of rRNA. So there are generally 2 subunits in the ribosomal RNA. So you can see the large subunit, which is the top portion here, and then the smaller subunit, which is the bottom portion here. And that's what's coming together to kind of create a sandwich for the mRNA to work in. So during protein synthesis, the tRNA is carrying the amino acid out from the cytoplasm to match up with the mRNA on the ribosome. And it's the tRNA matching up with the mRNA that's going to make the difference. And that's where the genetic code, and that's where the amino acids actually get identified. So the genetic code is actually read in 3 letter groups called codons. So you can see here we've got 1, 2, 3 codons. Okay, and those 3 codons, or those codons, are nucleotides that make up a word. So if you think about the genetic code, we have 4 different letters, or there's 4 different letters for DNA, which is A, T, G, and C, and there's 4 different letters for RNA, which is A, U, G, and C. So whether we're talking about 4 letters in our language. So those 4 letters are put together into 3-letter words, which is what a codon is. So that would be like saying that of the 26 letters of our alphabet, we're making 3-letter words out of those 26 letters, like the, cat, dog, that kind of stuff. And so that's what's happening here. So the codon is the word, right? And then the words are grouped together in the sentences, which is actually the proteins at the end of this. So that's how we get all of the different proteins and all of the different genes that are happening here. So you can see codon 1 would be AUG, that would be the word, AAC, UCU. Okay, and those are just the different, like I said, those are the different 3-letter words that we've made. What we do with those 3-letter words then is put them together to mean an amino acid. So we have this circle chart, which is really easy to use, okay? And then each ring in the chart is part of a different, or part of the codon. So the first letter of the codon is this center ring. Then the second letter of the codon is in this middle ring. The third letter of the codon is this outer ring, or the third ring. And then this outer ring is actually the amino acid that it codes for. Okay? All right. So you work in work your way in and move, or start in and move out. So if we were C, okay, the first letter of the codon, A would be the second letter of the codon, CA, and then C. So that would be our codon, and the amino acid would be histidine. And so that's the amino acid that needs to be put there. Now, one of the things that you'll notice, this third base, okay, that smallest ring around there, if you'll notice, many of the amino acids are coded for by more than one base in that third position. This third position is the very technical term of the wobble. That's the wobble position. Okay, because what that means is basically if there's a mutation in that position, and we're going to talk more about mutations in the next lecture, if there's a mutation in that position, sometimes it doesn't necessarily always have an effect, which is beneficial to us. There are some special codons that code for start and stop. Okay, so start is AUG, then methionine, okay, and then we have UAA, UAG, and UGA. Those are all stop codons. That basically means you've reached the end of the protein, stop, you're done. Okay. So what is this, how is this done? So translation, like I said, is step 2. Okay, so transcription was step 1, where we made this strand of mRNA. That strand of mRNA has left the nucleus, okay, and it's gone out into the cytoplasm or the endoplasmic reticulum and found a ribosome. So we're going to be focusing on translation. So if you're still confused about transcription, go back to the second lecture, or the last lecture, I should say, and you know, and kind of review that one. It's pretty short, but just keep that in mind. Translation, it's a little bit of a longer process. Well, not really a longer process, I should say. It's a little more of a complicated process, so bear with me. So during translation, what happens here is the mRNA actually attaches to the ribosome. Okay, so the ribosome, where the rRNA and the mRNA have come together. Okay, then the tRNA starts reading the mRNA and bringing the correct amino acid. So here we've got the codon is AAA. So the amino acid that codes for is lysine. So what's called the anticodon, which is basically the complement, the anticodon on the tRNA matches up and it says, oh, okay, here's the right one, drops off its lysine and moves on. Okay, and so what happens is the ribosome basically kind of chugs along the mRNA track, kind of like a train. Right? And what it does is it adds the new amino acids that match up. Once it's emptied, once the tRNA is emptied, it goes back off into the cytoplasm to get refilled again. Okay, and this just happens until it reaches a stop codon. Okay, so you can see it just moves along step by step, adding different amino acids to the polypeptide. So all these little balls here are amino acids. Okay, until it reaches this stop codon. When it reaches this stop codon, then it that tells it stop, you're done. Okay, and then it releases all of the parts, they all separate, and then the polypeptide goes off about its day. This is the central dogma of biology. The fact that DNA becomes RNA, which is then translated into a protein, that's what basically runs and makes every single thing in your body happen. I know it's kind of complicated to try and see it in this kind of lecture format, so there are going to be more resources, so just make sure you keep an eye out for them. But what's happening here is the DNA that's in your cell is being used, okay, to create a protein, which is then being used in the three-dimensional self that you are. So this is why you have whatever eye color you have, blue eyes, brown eyes, okay? This is why you have straight hair, curly hair. This is why you're tall, short. You know, this is basically everything. And then also, why is your body, or how is your body doing all of the different things that it's doing? All right, so this is a super important process to get right for your body to make sure your body runs. Now, when your DNA is actually being used to build a protein, that's when we call that it's being expressed. And that's when you're seeing that DNA, you're seeing that protein being made. And so when they talk about gene expression, that means that your DNA for that is actually turned on. And we're going to talk, the next unit is going to be all about genetics, you know, and all of that, Punnett squares and all of that stuff. So what's happening is, this is what's creating your features. Okay, this is what's creating your features. This is what's creating your the body, the three-dimensional space, the thing that's functioning. So translation in real, you know, if we were to use this as an example, okay, so this is the DNA strand from the last lecture that we transcribed into an mRNA strand, all right? And then we're going to turn that into a protein strand. So we divide it into three segments of three. So 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. And then we go read. So A, U, C, okay, is isoleucine. Okay, and then A, so that was AUC. All right. Now we've got ACC, which is going to be threonine, and then UGG, which is tryptophan, and then CUG is leucine. And then that would be the protein channel. So that's how it works. Okay? I hope this made sense, but like I said, there will be a couple more resources kind of looking at these two processes together, transcription and translation. So, and we'll talk more about that. All right? I will talk to you later. Have a great day.